Welcome, everybody, to our 10th Memory Decoding Journal Club of 2025. As you probably know by now, the Memory Decoding Journal Club is a bi-weekly event in connection with the Aspirational Neuroscience Prize. In particular, the Memory Decoding Challenge. That's a challenge to decode a non-trivial memory from static brain data. And today I'm going to be presenting the Nature Neuroscience paper Neocortical Synaptic Engrams for Remote Contextual Memories by Lee and co-authors. Recently, our Memory Decoding Journal Club has seen the presentation of some papers that we might describe as providing high-resolution data support for the standard model of contextual episodic memory. And this paper is a continuation of that trend. In fact, this paper uses many of the same techniques as the paper I presented on April 8th that was focused on memory updating by reactivating hippocampus from remote recall. And where we previously discussed mainly engrams, their reactivation and updating in hippocampus, specifically in CA3 and in dentate gyrus, we now turn to the consolidation of contextual episodic memory engrams involving hippocampal activity, but in their consolidated remote memory locations within neocortex. The slide that you're seeing right now is not from the paper. It's here mainly as a reference in case we need to discuss where the regions studied are located in relation to one another. You can see network connections from medial prefrontal cortex, their involvement in various behaviors, the location of the anterior cingulate cortex in mice and in comparison with human brain, and the mouse hippocampal circuit and the location of dentate gyrus therein, and the location of the mouse retrosplenial cortex. So you have some notion of where all these things are. My apologies. I am coming down with something a little bit, so I might cough from time to time. All right, so this is another fear conditioning case, just like in the previous paper. So all the mice were fear conditioned, and home cage mice had active neurons labeled during the home case conditions before fear conditioning after 2 days. And the contextual fear conditioning mice had active neurons labeled during the contextual fear conditioning. And fear conditioning behavior showed no significant difference between the two groups. In other words, they were all able, or at least learning was at approximately the same rates. The only difference is which group of cells was labeled. And the cells were then immunolabeled for remote recall. So here you see the setup there with 28 days between the contextual fear conditioning and the remote key recall task. And I'm not going to go through all of this in detail unless you guys want to, but this is, once again, the protocol they used for the labeling and how they used for OHT to open up a little window in which the labeling could take place and would result in, for example, showing the TD tomato labeled neurons wherever cells had been active. So there was what they found was there's a larger overlap of reactivated cells with previously labeled cells in the CFC group, which is as expected. These results suggested that CFC recruited medial prefrontal cortex, prelimbic, and basolateral amygdala neurons, which were more likely reactivated during remote memory recall than neurons active in the home cage. And 2.8% of the CAMK2 neurons, those are mainly the pyramidal neurons, so those that have that enzyme involved in LTP, in the medial prefrontal cortex 2 and 3 layers were among those labeled during the conditioning in the CFC mice. And if you look at these graphs here, you can see that as the 2.8 thousand. So this means that there were about 100,000 neurons in a cubic millimeter of mouse PFC. And of those, 6.5% projected to cells labeled in BLA, in the basolateral amygdala. They then tested direct reactivation of the medial prefrontal cortex cells' ability to induce the remote memory recall. And that was done by injecting channel rhodopsin and enhanced yellow fluorescent protein, and then carrying out optogenetic activation. This was done in a different context. This is what they're calling context B, and induced a freezing response. So the channel rhodopsin was expressed in the mice that exhibit the freezing behavior, and only the enhanced yellow fluorescent protein was expressed in mice that did not substantially exhibit the behavior. Did someone just ask a question, or is that just my, oh, that was just my chat. Okay, I should have turned that off. So you could say the reactivation of medial prefrontal cortex neurons active during CFC-induced fear in an irrelevant context. That's what they were able to show. And their results suggest that a subset of the medial prefrontal cortex neurons active during CFC was reactivated during remote memory recall, and optogenetic reactivation of these neurons induced memory recall. And they then gave those a name. They call these medial prefrontal cortex neurons the medial prefrontal cortex engram neurons. Next, they wanted to know if remote consolidation strengthens connections between medial prefrontal cortex neurons. And as we go through this, they're going to do this first for interhemispheric connections, and then afterwards for local connections between these MPFC neurons. So they injected a virus, the AAV-DIO-CHR2-EYFP, into the medial prefrontal cortex of the CFC-labeled mice. That was done unilaterally. Brain slices were collected after remote recall testing in order to carry out EFIS recordings. And the expression of the CHR2-EYFP allowed them to identify axons from the injected cells in medial prefrontal cortex that innervated the contralateral MPFC. So the ones that were innervating there, they call those the engram inputs, and those were sparsely distributed. They then carried out patch clamp in MPFC, which showed that the EPSCs, so the postsynaptic currents, during photostimulation of the engram inputs. The contralateral engram input neurons showed EPSC effects from the activated engram neurons. In pharmacological isolation, AMPA receptors activate at a more hyperpolarized clamp potential than NMDA receptors. That allows them to record EPSCs for each type of receptor monosynaptically by using voltages of -80 millivolt and +40 millivolts respectively. Compared with the engram to non-engram synapses, engram to engram synapses displayed larger AMPA receptor-mediated EPSCs. Also, the ratio of AMPA to NMDA EPSC amplitudes was higher in the engram to engram synapses than in the non-engram to engram synapses. So interhemispheric MPFC engram inputs were more strongly connected to MPFC engram neurons than to non-engram neurons 28 days after the contextual fear conditioning. This was also observed after 14 days, but it was not observed after only 7 days, which to them suggests gradual strengthening during consolidation. The consolidation did not depend on induced remote recall, as the same was recorded without the remote recall phase. Furthermore, silencing of remote MPFC engram neurons did inhibit behavior after 28 days, but not after 7 days, indicating when the behavior depends on the MPFC engrams. They then examined if consolidation-related synaptic strengthening was input-specific. The CHR2-EYFP was globally expressed in the CAM kinase 2 pyramidal neurons in the AAV-injected MPFC, so the part of the MPFC in which they induced activity. Engram neurons were only a small subset of those. Most of the axons were non-engram inputs. Photostimulation induced EPSCs from both sets, 4 weeks after the contextual fear conditioning, photostimulation of those axons induced EPSCs in the tomato engram and also the tomato non-engram synapses. So the non-engram to engram and non-engram to non-engram synapses in contralateral MPFC. The AMPA-mediated EPSCs, as well as the AMPA-NMDA ratio, did not differ between the non-engram engram and the non-engram non-engram synapses. This suggests that non-engram inputs to MPFC engram neurons were not strengthened. And therefore, remote memory consolidation involves synapse-specific strengthening of excitatory connections between MPFC engram neurons. And they then termed that whole thing the MPFC engram circuit. Next, they wanted to know if system consolidation also strengthens engram circuits in the caudal ACC and the retrosplenial cortex, both of which have been implicated in the consolidation and retrieval of remote contextual memories. Engram to engram synapses exhibited larger EPSCs than engram to non-engram synapses after 28 days in the caudal ACC. Engram to engram synapses did, on the other hand, not exhibit larger EPSCs than engram to non-engram synapses after 28 days in the retrosplenial cortex. So it seems that some neocortical engrams are more likely than others to undergo strengthening during consolidation. Then they wanted to take a look at the CREB dependence of the MPFC engram circuit. CREB, the camp response element binding protein, plays a crucial role in neural plasticity by regulating gene expression. Essentially, CREB helps to mediate the long-lasting changes in the brain that are the basis of learning and memory. CREB inhibition, in MPFC engram neurons has been shown to prevent remote memory consolidation, and so they went ahead and inhibited CREB in MPFC neurons after contextual fear conditioning. Photostimulation was used to induce EPSCs in mutated CREB engram, that's the engram to engram synapses, dominant negative mutated CREB engram, also on engram to engram synapses, as well as dominant negative mutated CREB on non-engram neurons. And the AMPA to NMDA activity ratio on dominant negative mutated CREB engram neurons stood out as different from the rest, while mutated CREB expression on MPFC neurons prevented strengthening. So the CREB in MPFC engram neurons is indispensable for the strengthening of the MPFC circuit during systems consolidation. They then examined the effects of extinction
Sorry.
of remote memories. Yeah, go ahead.
Can you, either I phased out, I missed it. Could you jump back one slide? I'm just trying to remember the timeline of when the CREB.
Sure.
Do you remember when the mutant CREB was administered relative to when they did the training? I'm not sure it says there.
Does it not say?
Or is it immediately after CFC or Drew?
I believe it was right after.
Yeah. All right, cool. I can check that.
I'm seeing it correctly. Yeah.
Thanks. Sorry.
Yeah, no worries. All right, so we'll carry on with the extinction test. They wanted to look at the effect of extinction on remote memories. And they did this by carrying out repeated exposure to the same threatening context, so the context A, without applying the foot shock. And that was done for 5 days, 4 weeks after the initial CFC training. And indeed, freezing behavior decreased, so the memory was being extinguished. And in that case, AMPA to NMDA ratios no longer differed. So extinction weakened the previously strengthened engram to engram synapses within those engrams. The results suggest that remote memory consolidation involves the strengthening of interhemispheric connections between MPFC engram neurons. So this was the interhemispheric part. Next, they explored the local excitatory connections. Yes,
I'm sorry, I have a quick question on the previous slide.
sure, no problem.
Sorry. So that was done, the extinction was done 28 days after. So it was having to be a remote, it had to be on the remote memory that was was being extinguished.
That was the idea. Yes.
Do you happen to know if they were to do that at like 7 days or something? Would that uh, essentially create a new hippocampal memory or something? Or
Hmm. I mean, I don't know for sure, but it wouldn't surprise me if you would be setting up a new hippocampal memory under the new condition.
yeah, it just
But you
strikes me
could,
that
yeah,
the extinguishing a memory at that late of time might be very different from extinguishing at an earlier time. In any case, just
yeah.
wanted
I don't.
to,
That's why I was momentarily just kind of nodding, sort of putting my head aside and wondering, wait, is that extinction, or is that because you haven't consolidated yet? Is this just introducing a new context? Another one. Anyway, yeah. So I don't know.
hmm.
Interesting
I guess if
question.
we're discussing, yeah, I found this fascinating because I've not looked up extinction or learnt much about it for a long time, but I always had this vague memory that like extinction is not the same as forgetting. It's like learning a new memory to suppress the like previous one. And that here in this paper, they're talking about how during the extinguishing, they actually see like a weakening and like a return to baseline of the actual circuit that like is involved, at least one of the circuits involved in this. And I was like, oh, that's different to vaguely what I remember having learnt about previously. So that was cool. I don't have a question beyond like it was an interesting difference to
That's
what
interesting
I previously learnt
observation.
about.
Yeah. Yep. All right. So moving on to the local MPFC circuit. And that's what they wanted to explore next. So here, photostimulation of the CHR2 engram neurons induced EPSCs recorded in adjacent engram and non-engram neurons in the ipsilateral medial prefrontal cortex. And they then carried out quantal measurements of the EPSCs in engram neurons and found that they were substantially larger than in non-engram neurons. Local engram to engram synapses were therefore strengthened during consolidation. This was not apparent after only 7 days. The effect was also observed without inducing recall, so this was due to gradual consolidation. Well, I didn't put very much on the slide here for this, but they also examined inhibitory engram inputs to medial prefrontal cortex pyramidal neurons, and the GABAergic engram inputs to medial prefrontal cortex engram neurons were substantially weaker than those inputs to non-engram neurons. Thus, MPFC engram neurons received weaker inhibitory engram inputs than other neurons. I was actually slightly surprised by this, but maybe it's just because I haven't been keeping up with the literature on interneuron plasticity during this process. Or the input from interneurons anyway. So then a look at dependence on dentate gyrus, because dorsal dentate gyrus has a critical role in MPFC engram maturation during system consolidation. And they used a genetic method to ablate the dentate gyrus engram neurons that are active during contextual fear conditioning. After ablation, freezing behavior was reduced. So either consolidation or retrieval was inhibited by the ablation. And CFOS labeling in MPFC and basolateral amygdala indicated reduced reactivation of engram neurons during remote memory recall when dentate engram neurons were ablated. But they went on a little bit further with this one. So this raises the possibility that sustained activity of dentate gyrus engram neurons may strengthen the medial prefrontal cortex engram circuit and facilitate MPFC engram maturation. So in this this ablation group, the CASP3 group, they found that the peak amplitude of evoked quantal EPSCs didn't differ between engram versus non-engram neurons, and that indicated an absence of strengthening of the local MPFC engram circuit. Those results indicated a critical role of dentate gyrus engram in the strengthening of MPFC engram circuits. So sustained activity of dentate gyrus engram after learning can contribute to remote memory consolidation, possibly by strengthening MPFC engram circuits. And the ablation of dorsal CA1 engram neurons active during CFC prevented the strengthening of local MPFC engram circuit.
I'm sorry, Randall. Can I can I ask about the timing of that? So the ablation was at what time?
I don't remember exactly, but it was carried out in connection with the CFC. So shortly after contextual fear conditioning.
Okay, so presumably, if this had been done like 3 weeks afterwards, it would not be hippocampal dependent.
No, this would have to be very shortly afterwards. Yeah.
Okay. Okay. So this really is not talking about the consolidated memory.
It's talking, in my opinion, about the ability to establish a consolidated memory. So the reactivation via dentate gyrus during this, when you have those sharp wave ripples going on to do consolidation in outside of hippocampus.
Okay. So did they show that the memory was formed and could be retrieved in the hippocampus before they did the ablation?
I don't believe so, or at least I didn't see that in the paper.
Okay, because I mean, if they just wiped out the hippocampus, then this would not be.
Yeah, I got the lines. Yeah, so in this group, dentate gyrus engram neurons, blah 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 blah, underwent the caspase-3 mediated cell death, efficient ablation, 28 days, but not 3 days after CFC. 1 day after CFC, mice showed robust freezing behavior, while mice in the CASP3, displayed much less freezing at the 28-day later mark. So at the 1 day mark,
did you just say that they only ablated 28 days after?
No, they say that the ablation takes a while to kick in. So like, it's saying that 3 days after CFC, the neurons are still alive. It's only at 28 days later that they're dead. And that the mice are still showing freezing behavior at the next day, like 1 day after CFC, and then they lose it by 28 days in the ablation group.
That's a gradual process there as well, yeah.
My reading, yeah.
Okay,
they got the little image for it as well. It's like in 5C or something where they show the, So yeah, I think they're saying the memory is still formed initially, like at 1 day. It's just not there at 28 days. If that answers your question, Ken.
Well, I think it does, but I want to be very specific. So this ablation of dentate gyrus neurons is messing with the consolidation process. That I think that's what you were saying, Randall.
Yeah.
Okay, so it's stopping, and presumably it's stopping in like a week. In the week to 2 week type of range, the type of reverberant activity during sleep or whatever that would.
I think that's what the line that Ariel just read out would say. Yeah.
Cool.
I guess a variant, and maybe this is what Ken's getting at, is also what would be interesting is if you left those neurons alive, but you blocked their ability to project onto the MPFC or something, where like maybe, at like day 2 or day 3, the mice could still express the memory somehow due to the presence of the hippocampal neurons, but they couldn't consolidate it because they couldn't project to MPFC or something. And like, we don't have the data for that, at least from this experiment, or at least this particular part. I can't remember if they do that in a later section. Whereas here, the CA1 neurons are just kind of alive for a while and then they die off at not really clear exactly when they die. Somewhere between 3 days and 28 days.
Okay. Yeah, but we don't have that data in this paper. No. So we can't see the alternative. Anyway, they do carry on a bit with the CA1. So one thing they point out is that the dorsal CA1 only weakly projects to MPFC. And so there may need to be an intermediary or different kind of transport, another route. And so they examined presynaptic inputs to MPFC engram neurons. And you can see a whole set of them lined up here, various options. M-cherry labeling showed the inputs from the caudal ACC, from retrosplenial cortex, lateral entorhinal cortex, ventral CA1 hippocampus, and basolateral amygdala, and showed that there were only very few connections coming in from DCA1, but many other options here. They then examined if the retrosplenial cortex may be relaying the dorsal CA1 engram activity to medial prefrontal cortex engram neurons. And they did this by photostimulation of the DCA1 engram neurons and showed that this caused monosynaptic EPSCs in the M-cherry labeled RSC neurons. And so RSC neurons projecting to MPFC engram neurons received monosynaptic DCA1 engram inputs, and that suggests that a subset of RSC neurons could convey the signal of DCA1 engram to MPFC engram during remote memory consolidation. Ablation of engram neurons in DCA1 or in RSC, or in the retrosplenial cortex, inhibited remote memory recall, which seems to support this memory consolidation circuit.
So Randall, the retrosplenial cortex, there's other literature on that had I remember somewhere, I haven't looked it up, but there is some other literature that says that retrosplenial cortex is used for the consolidation circuit. Do you happen to know that other research? Because they're not pulling that out of this, these 6 regions, let's try this one. There's like a set of literature that says this RSC is important for
yeah,
remote.
I was wondering why they picked on that one particular, but I didn't previously look at the other literature for that because for some reason the pathway from CA1 into PFC in terms of all its potential paths or routes was not something that I have studied before.
Okay, so I think, I mean, I shouldn't say anything, but I think there was some paper that directly inhibited that or knocked it out and showed that it would prevent remote memory consolidation.
Okay, so that's why they picked that and confirmed, at least somewhat confirmed that possibility. Yeah. All right, so here they refer to previous studies that suggested a critical role of the MPFC basolateral pathway, basolateral amygdala pathway in the recall of remote contextual fear memories. And what they did is they found that silencing of MPFC neurons projecting to the basolateral amygdala prevented the recall of remote, but not recent contextual fear memory. 4 weeks after CFC, photostimulation of the CHR2 medial prefrontal cortex engram inputs induced EPSCs in tomato BLA neurons, indicating that MPFC engram neurons monosynaptically projected to BLA engram neurons. And both the AMPA EPSC amplitude and AMPA NMDA ratio were substantially larger in the tomato BLA neurons than in those not tomato labeled, suggesting stronger, not activated, well, they were labeled, of course, suggesting stronger connections of MPFC engram inputs to BLA engram neurons than those inputs to non-engram neurons. Only a small fraction of MPFC engram neurons project to BLA, so they wanted to know if there might be PFC relay neurons involved. And they found that MPFC engram neurons can activate the BLA engram neurons through MPFC relay neurons and contribute to remote fear memory recall. During remote memory recall, reactivated MPFC engram neurons may recruit other MPFC neurons that were not active during the contextual fear memory conditioning, and they now call those the MPFC recall neurons. I'm not sure why they didn't go with relay neurons, but they call them the recall neurons. Using dual independent labeling, they independently labeled MPFC neurons active during CFC and those recruited during remote memory recall. And photostimulation of the CHR2 engram inputs induced glutamatergic and monosynaptic EPSCs in the tomato CHR2, tomato and not CHR2 MPFC neurons, indicating that the MPFC recall neurons received excitatory inputs of MPFC engram neurons. Moreover, the AMPA EPSCs and AMPA NMDA ratio were substantially larger in those neurons than in the non-tomato, non-CHR2 neurons, or tomato minus CHR2 minus neurons, indicating that MPFC recall neurons receive stronger MPFC engram inputs than other neurons did. So, during remote memory recall, reactivated MPFC engram neurons can efficiently recruit MPFC recall neurons, and consistent with that, silencing of MPFC neurons that were active during remote but not recent memory recall inhibited subsequent memory recall or remote memory recall. What you can see here is this depicted as a model that on the left, some of the neurons recruited during acquisition are recruited during acquisition and they're weakly connected to one another initially. Then during consolidation, that strengthens the connections between those engram neurons. This is the local engram. And on the right, you see that recall activates some engram neurons and some relay neurons. So the contextual memories gradually mature to a stabilized form in the neocortex. And in closing, kind of as a summary, so after system consolidation, the retrieval of remote contextual memories requires neocortical activity and depends less on hippocampal activity. During system consolidation, MPFC engram neurons slowly undergo enduring neuronal and synaptic changes for long-term memory storage. In this study, they demonstrated that the long-term storage of remote contextual memories involves progressive and synapse-specific strengthening of excitatory connections between medial prefrontal cortex engram neurons. Both the interhemispheric and local excitation connections between MPFC engram neurons display higher synaptic efficacy than those between engram and non-engram neurons, or those between non-engram neurons, suggesting synapse-specific strengthening of MPFC engram circuits during system consolidation. The strengthening of the engram circuit was also observed in the anterior cingulate cortex, but not in the retrosplenial cortex, although both areas are involved in remote memory consolidation. The strengthening of MPFC engram neurons, engram circuits was not detected 7 days after CFC, indicating that the engram circuits were progressively strengthened during system consolidation. And the study highlights the role of sustained activity in MPFC engram maturation, as MPFC activity is modulated during hippocampal sharp wave ripples. Hippocampal signals can be conveyed to the medial prefrontal cortex, facilitating hippocampal neocortical interactions during sleep for memory consolidation. And the retrosplenial cortex can convey hippocampal signals to MPFC engram for system consolidation, according to the measurements they made. Although the medial entorhinal cortex is necessary for MPFC engram generation, activity in the medial entorhinal cortex to MPFC pathway is not required for remote memory recall. During remote memory recall, inputs likely activate only a fraction of MPFC engram neurons, but excitatory connections between MPFC engram neurons were strengthened during remote memory consolidation, while MPFC engram neurons also received weaker inhibitory engram inputs than other MPFC neurons. And so this can help reactivate the whole engram. While a subset of MPFC engram neurons was reactivated during remote memory recall, other MPFC neurons that were not active during initial learning were also recruited during remote memory recall. And that puts together the whole picture of what they have shown us by studying the circuit in such great detail. And that is that. We can discuss. Ken, did you happen to go back to the go-to paper and find the timing there?
I did. The go-to paper shrinks everything down to like 2-3 days type stuff. So I don't know if it's relevant. But what the go-to paper did is they had a technique called Cali, or a particular type of Cali that would, if they shined light on synapses, they could shrink those synapses. And it was only those synapses that were undergoing reactivation, essentially. And so at the 3-day mark, I should look this up here. I had it right here. They said Cali in the anterior cingulate cortex that was triggered on day 2 would erase a memory that was tested on day 3, which is just crazy. Okay, so that's kind of saying that you can erase that. That means that it's out of the hippocampus by day 3.
That's very
okay,
fast.
so which this paper is at odds with, and I guess a bunch of other papers. Maybe the go-to paper doesn't make sense, because it seems like the timing is off by many days in terms of when it's hippocampal dependent, when it's.
Let me just check that I really understood this correctly. So they did at first show that the memory had been consolidated before they then erased, or was it just that they made it impossible to consolidate?
Made it
Sure that they
impossible
meant
to consolidate.
okay?
I think that's what they were. That's what
But
they
I can
were.
imagine. I can imagine that you can keep consolidation from happening, starting at a very early time, on a very early day, right?
Absolutely, but they did the testing at day 3. And so if it sounds like if you were doing testing at day 3, that you wouldn't, that it would still be hippocampal, that the hippocampus would still do its job. Maybe that's not the case. And there's
Hippocampus.
a factor here that I can't remember the paper, but there was some paper that was, and it might have been the retrosplenial cortex paper, that was kind of asking the question, if you have a memory that is in the hippocampus and you have a memory that is in the anterior cingulate, or in the frontal cortex, let's say, in the cortex,
Yeah.
Then what decides which one to use? And
Also,
it's not necessarily.
can we back up one step?
Yeah.
What does it even mean to have a memory only in the hippocampus? Can the hippocampus retrieve any memory without help from other areas of the brain, from neocortical regions being involved at all?
I was assuming that was the case, at least for the frontal cortex, because isn't the idea that the frontal cortex.
No, that's fine. That's fine. So I just want to make sure I understood what the go-to paper did, because maybe I wasn't paying enough attention. But,
yeah, I mean, it's a paper that is just as long as this paper, and this paper is really long.
Yeah.
So there might be some details that I'm not remembering correctly, but essentially the go-to paper was kind of saying,
Where
yes.
where did they break the pathway? Where did go to inhibit, remove,
So the
ablate, break?
thing is that they were never killing off neurons or inhibiting neurons.
Yeah.
Okay, they were doing long-term, relatively long-term Cali. So this to wipe out those synapses that were undergoing LTP over a several hours period. And so they
Should we really
identified,
be distinguishing between wiping out synapses or killing neurons when it comes to breaking a pathway?
That was in the cortex that they were wiping them out, or where were they wiping them out again?
they did both, but what I'm talking about is in the cortex.
Yeah.
So I guess the reason why I'm saying that is that you can imagine if you were just inhibiting a pathway, then that might stop a retrieval. But if you're they were at some time point shrinking synapses and really trying to identify when the learning was occurring. The whole point was to identify when the learning was occurring, and using this technique in order to kill those synapses that were undergoing learning in a particular window. And they could say if on the second day's sleep periods you killed off all the synapses that were in the frontal cortex that were going to be consolidating this memory, then you wouldn't be able to retrieve the memory on day 3. And so that says two things to me. It says that that there was that the hippocampus was training during that time period, and it says that that at day 3, those newly trained frontal cortex neurons were the ones that were being used for the retrieval. Now, in this paper, it seems like that is shifted way out, so that the hippocampus is over a full week training these synapses in the medial prefrontal cortex and the anterior cingulate, and it's only after that full week that those are going to be mature enough to support a memory on their own.
Right. So the extinction stuff is the main place where I would have a concern about a something not aligning between the go-to paper and this one, because I can understand, because in the rest of the paper here they are looking at the strengthening of synapses over time, and in the go-to paper you're looking at killing synapses. So you could imagine that even if after 7 days you don't have sufficiently strengthened synapses yet to have a remote memory recall, that doesn't run counter to the possibility that killing synapses 3 days after or 2 days after could prevent consolidation. So those things are not in opposition to one another. But maybe this extinction thing here might be a problem.
So I think my key question is something simpler. Over what period of time is the hippocampus, does the hippocampus still have the memory? And over what time period does the cortex have the memory? And is there a switching between the two?
But do we have that data from those two papers, from this one and from the go-to, or do we need to get that from somewhere else? Because I don't remember if the go-to paper, if they test hippocampal ability to recall after they've done that killing of synapses in frontal cortex.
I think they do, because what they're saying is that on day 3 they test, and the mice
But
don't.
this isn't a test purely of remote recall. It's a test of
That's
recall
right.
at all.
It's a test of recall at all, exactly. And you would think
Ooh, this
that
is very
A,
short.
it doesn't. Yeah, exactly.
Can I
Yeah.
just to check something? I may have forgotten this, and it may have been done in one of the papers we discussed. But if you, and this might be exactly the go-to, what the go-to's paper is talking about. If you destroy those cortical strengthened synapses, so like you weaken them or you prevent them from strengthening, but you hold on to the engram in the hippocampus and you forcibly reactivate the hippocampal engram, do you see like freezing behavior, or is the freezing behavior blocked? Um,
Yeah, that's kind of what I was just asking Ken as well, which is, do they just test recall at all, as in the freezing? And Ken is saying yes, they did. So they didn't see the freezing anymore after killing it after 3 days in the frontal cortex. But so then another issue that could be involved is how the memory is supposed to be reactivated in hippocampus. So how that signal is reaching it and whether it's going through potentially any of the areas where they did the synapse killing, but I don't think they did, right? Like this is just for just where consolidation was happening, not the path, for example, to dentate gyrus or something like that.
So we've beaten the go-to paper to death. I have other questions.
Okay. All right. Even though we don't yet know the answer to how come that can happen.
Well, along what you were just saying, though, I have a question on that. So, I was wondering. So the remote memory. Maybe you just asked this, or but when a remote memory at 28 days is being recalled, what are the inputs? I guess the idea is that it can't be hippocampal dependent because you can wipe out the hippocampus and it'll still be there. So then what are kind of the inputs? Are the inputs cortical direct to prefrontal?
Well, we know that the medial entorhinal cortex is not needed to get there because you can remove that and still get the remote memory recall to happen. Gosh, what is the other pathway? Did they not test that? I thought this must have been part of this test, this whole series of tests.
When you say it's not the medial entorhinal, was that an experiment that they did here?
They didn't do it. They merely remarked upon the fact that was a known thing, that you don't need MEC. Where do they have this?
I mean, this is contextual fear memory, so at some point you have to have some area of the brain that is representing the context, the cage.
Right. Yeah. No, that's clear. So the question is, what pathway? Gosh, maybe they didn't do this part. Yeah, that might not be in here.
I mean, why I'm asking all this is because I kind of feel like there's a ton of stuff in this paper, and what I'm kind of missing is this overall picture of here is all the pathways and all the timings. Maybe it's too much to ask for this paper, but this there's a lot of papers that have addressed this.
It would be really nice to have that, just kind of a sequence from acquisition to consolidated remote memory recall, and what are the pathways during acquisition, what are the pathways after consolidation, and it seems like we're still missing a little connection here from I wonder if we can't possibly pull that out of these diagrams, can we? No, we can't. That is not sufficient.
Yeah, I don't even know off the top of my head if you ablate the hippocampus, you've got the memory in the cortex, but you test at 28 days or later. Is it still remain context specific, that memory, or in the absence of the hippocampus, does it get a bit non-specific to the ability to bind like the various different cage aspects together into like that's what I found scary. In that actually this is a slightly ill-formed question. Other people can ask questions. Let me think about how to rephrase this.
The other thing I thought was very cool was the frontal cortex connections to basolateral amygdala. And is there a distinction there that this is kind of, this is not lateral amygdala, this is basolateral amygdala? It's kind of like lateral amygdala would be, I've learned a sensory memory, like an auditory memory, and then maybe it activates basolateral amygdala to do the actual freezing. And so lateral amygdala looks like the same thing as a, so this would be the medial prefrontal cortex engram would be a full-on memory, just like you would get in the lateral amygdala for a sensory memory.
That's possible. Also, I think back to our previous question about how the retrieval would happen in remote recall in the absence of hippocampus. I believe that is where the connection through the anterior cingulate cortex may play a role. I was just doing a quick search about that. And they did, of course, test the connections, the engram connections being formed between MPFC and ACC. That was one of the things that they looked at here. Although I'm trying to find this, I'm having trouble finding the slide right now. I don't remember where that was. Maybe this was after this one. No, I'm having trouble finding it right now. But I think ACC might have been part of that pathway.
Should we spend a minute wondering what implications this whole work has, if anything, for memory decoding? Just given it's the aspirational neuroscience.
What are your thoughts?
I think this is a really cool paper that informs my understanding of how memories are formed in, the hippocampus and the cortex. But on reflection, I'm not sure what to do with it in terms of anything directly informing extracting memories. I mean, it's like, I guess the implications are there's that specific engram to engram strengthening of synapses in the cortex that are underlying these specific memories. But we kind of already knew about that from other papers we've looked at. And I'm not sure how you'd go about trying to identify them or tag them or somehow link them up to specific memories. What else is there? I don't know, did anything else stand out to anyone?
I'm not so sure that we knew this. So the thing that stands out to me on this is that, and maybe this is somewhere else, but I didn't see it somewhere else, is that they are saying that the medial prefrontal cortex engram cells are interconnected, that there's engram to engram cells. And that to me sounds like an attractor that could be extracted, right? And so a theme that has been going through, that may be wrong, but a theme that has been going through is that every memory is associated with a particular ensemble of interconnected excitatory cells in a particular area. And that would be the thing that you would reach into and say, there is this many memories, long-term memories, if we were to look in medial prefrontal cortex, as opposed to looking in lateral amygdala and saying, there's this many sensory memories. Or if we were to look in CA3, let's say, I'm not so sure about that, but to say there's this many very recent memories. But all of them would have an attractor, an ensemble of interconnected neurons associated in a one-to-one fashion with those memories.
Yeah, the one thing that I found a little bit, I guess, unfamiliar in the way that the engrams were being described as MPFC to MPFC neuron connections was that I had always been thinking about prefrontal cortex in terms of these, mini columns or micro columns through multiple layers, having a fairly kind of standardized circuit or subcircuit within each, and then creating connections between various mini columns or micro columns, and that therefore you have this sort of very standardized, somewhat higher level circuit that's building useful memories or codes or programs in a sense. And now I'm wondering about taking this very cell to cell view and comparing that with the columnar view. And then what would you actually want to identify? What would it really map on? Does this map onto these columns? And should I be thinking of it more like an engram as this connection between these microcolumn structures, or what? Yeah.
Yeah. Yeah. I mean, I think a different way of saying that is do these cells have some kind of feature representation outside of just being a collection of cells that redundantly encodes a particular memory?
Yeah, because the structure there is completely different than what you would find, for example, just between cells in CA3 or something like that.
Yeah.
So one bit that's missing that I would like is if we had a neocortex paper like this that actually gives us, once again, some structural reconstruction from electron microscopy, for instance. Well, that's a very different kind of paper than this one.
Is there an appetite for doing any of the microns papers, or there's a cortical column paper that's, I believe, still in bioarchive from the Helmstetter lab that is very, very detailed in mouse barrel cortex.
I mean, personally, I would like to see another microarchitecture paper.
Yeah, I'd be keen. Is that purely microarchitecture, or is it like linking to function or memory or behavior or something? And like, what do they do in the Helmstetter one?
I don't think they have any recordings alongside it. They certainly tie it back to other literature. But yeah,
that's more than enough for me.
but it is a bioarchive, so
Is that a no-no for our? It's not a no-no for our journal club, is it?
It's definitely not a no-no for the journal club. It may be a no-no for an aspirational neuroscience award.
Right. Yeah.
Fair enough.
It's hard enough to, I have to say, I mean, some of the aspirational neuroscience awards that we've already given, like the GoTo, are falling into the category of, was this too was it too soon? Should have been looked at in context of the full literature. It is really, really hard to, it's hard enough to get through a paper like this. You did great, Randall. That was fantastic. This is just an insanely long paper. But it's hard enough to read this paper, let alone put this in context of the wider field and say, they're getting something that's way out of the norm, or they're getting something that was already known 20 years ago.
But yeah, I would like to see that paper come up at some point. So if you don't mind dropping it into the spreadsheet, then we can schedule it somewhere.
Great.
Yeah. I'll even volunteer to present it, I guess, because,
Will do.